All right, in this PowerPoint, we're going to take a look now, or start taking a look at our gram-negative bacteria. So we've gotten through all of our gram-positive, now we're moving on to our gram-negative bacteria. Now, before I move on too much, and I'm like, there are two terms to kind of go over, and the difference, because a lot of these can cause infections, is knowing what a foodborne intoxication is versus a foodborne infection. A foodborne intoxication means that you're going to have some type of food sickness because of the toxins that the bacteria made. That it's not the bacteria that are actually causing problems, but it's the toxin that those bacteria make that actually causes you to have those food poisoning symptoms. Foodborne infection means you ingested live bacteria from food and they grow inside your intestines and cause you to have food poisoning. Now, why the difference? One is you're ingesting live bacteria, one is that you're ingesting the toxins, is because if you have bacteria that are growing in food, like you always hear, well, make sure you cook your food properly or freeze it or refrigerate it. Well, cooking your food properly can kill all the bacteria. But if they, while they were in the food, already made toxins, the toxins may not be damaged from that cooking, which means you can still get food poisoning from eating, and I'm like, food that toxins or bacteria lived in. Even though you killed the bacteria, the toxins are still there. So even though your food is cooked properly, you have to look to see how was that food handled prior to cooking. If it was sitting out for a long period and there was lots of bacteria in there producing lots of toxins, even if you killed all the bacteria, you may still get foodborne intoxication, meaning the toxins are still going to cause problems. Only because a lot of these bacteria in our gram-negative group cause food poisoning type symptoms. Now, our gram-negative bacteria is our largest group of human bacterial pathogens, partly because gram-negative bacteria have that lipid A in their lipid A in that outer membrane. If you remember, gram-negative bacteria have an outer membrane and an inner membrane, sandwiched around that really thin cell wall, that really thin peptidoglycan layer. And that lipid A has lots of antigens and lots of virulence factors, meaning it is more likely to cause disease. And it usually, when it gets broken down and released, triggers fever, vasodilation, inflammation, shock, and then DIC, which stands for disseminated intravascular coagulation, aka you get blood clots, which is never good. Now, just like gram-positive bacteria, we are going to group our gram-negative bacteria first by shape. So we've got gram-negative cocci and gram-negative rods, and I'll hand you this flow chart out in lecture. And I'm like, from there, if you notice gram-negative cocci, there's one group. That's it. One genus. Gram-negative rods, though, there are a bunch. And the way we're going to start to break down our gram-negative rods is by their oxygen requirement. Whether they're facultatively anaerobic, which means they can really go either way. And I'm like, they can grow in either, whether they're straight aerobic or straight anaerobic. But even of our facultatively anaerobic, there's a lot of bacteria here. And so then we're going to group them based on a test we can do in lab called an oxidative test. And we'll do an oxidative test in lab. One group is oxidative negative, one group is oxidative positive. My little note, even though this test is used to divide up our facultatively anaerobic, that doesn't mean any of these other bacteria couldn't be oxidative positive or negative. It's just a test we use to divide these guys up. And so, like our gram-positive, we're going to start on the left and work our way over to the right. So we're going to start looking at our gram-negative cocci group, which, yes, there is just the one. So it is the easiest. If you are seeing gram-negative cocci bacteria, you are in the Neisseria group. Some of you saw this in your throat. You ID'd it down. You're like, oh, yes, I have gram-negative cocci. You've got lots of normal flora in your throat. And I'm like, there are lots of normal flora that are Neisseria. And I'm like, now most Neisseria like to hang out in pairs. So they're diplococci. They don't swim around. They're non-modal. There's no flagella on them. And I'm like, but lots and lots of normal flora. If we did test them, they'd be oxidative positive. However, in this genus, there can be some pathogenic species as well. So lots of normal flora, but there's some pathogenic ones as well. The two main pathogenic species in our Neisseria group are Neisseria gonorrhoeae, it's like two E's in there, gonorrhoeae, and Neisseria meningitidis. Now, in general, and I'm like, we're going to start with the Neisseria gonorrhoeae, which causes gonorrhea. And it's these, again, diplococci, that like to stick to the genital region, the urinary region, and the digestive tract. In men, it causes painful urination and pus, which is usually what sends them to the doctor. In women, it's usually asymptomatic. Now, my little note about that can actually become a problem because women may actually be carrying this bacteria with no idea, which means they could be passing it. This should be in blue too. It can, however, sometimes trigger pelvic inflammatory disease or disorder, but a lot of times for women, asymptomatic. In newborns, however, because a woman may be asymptomatic, have no idea they have it, and then they give birth. And if it's in their genital region and they give birth, this is a top cause of eye infections in infants. Now, luckily, the trend for this particular bacteria is going down. A lot of this, there's a lot more testing. They test females when they're pregnant. And I'm like, there's more routine testing for this particular bacteria. There's a lot more news out there, more information out there of wearing condoms, safe sex, all of that stuff, that the number of cases are going down. Now, diagnosing someone for Neisseria gonorrhoeae, they can do genetic probes. They can look for the genetics of this particular bacteria, especially if asymptomatic, if you're not exhibiting any symptoms. What they're really looking for is, what they're looking for other than genetics, they can also do a gram stain on these bacteria. And I'm like, these bacteria like to live inside of other cells. They can get ingested from white blood cells, and then they'll live inside of them. So they can start to see some of these white blood cells. And then inside of them, we've got these gram-negative cocci. Gram-negative cocci, right in itself, there's only one group of bacteria that are gram-negative cocci. But if you also see them intracellular, it's a huge clue on, what bacteria they are. Now, treatment, unfortunately, they're starting to get resistant to a lot of different antibiotics, but they can do a broad spectrum antibiotic. They usually do it intramuscular to really get that high concentration right away. And a lot of times they're also doing a combination of antibiotics to really try to kill any and all that might possibly be there. Best idea is just to not get it in the first place. Abstinence, condoms, testing mothers, otherwise newborns, a lot of times will have antimicrobials put in their eyes right at birth if the mom tests positive. Now, the other Neisseria that can cause problems is Neisseria meningitidis. Now, it is normal flora of the upper respiratory tract, so it's not uncommon to find this in the upper respiratory tract. However, it becomes life-threatening if it gets somewhere else. If it gets in the blood or the cerebral spinal fluid, this is when it becomes deadly. Now, it can be transmitted by respiratory droplets in closed areas. So people that live in closed areas, they live in dorms, they live in various schools, and I'm like, or in schools, they live in barracks if they're in the military. And I'm like, because the two diseases that it can cause depends on where it gets, whether it gets into the blood or whether it gets in cerebral spinal fluid. If it gets into cerebral spinal fluid, it causes meningitis. And I'm like, Neisseria meningitis, or bacterial meningitis. There's lots of things that can cause meningitis. And I'm like, but if it's the Neisseria that can cause it, the highest risk are those that live in closed areas. There was just a big, huge outbreak. A couple students got this, which really is an outbreak, at UW-Madison, the fall of 2016, so about a year and a half ago. There was a couple students that got it that lived in the dorms. And I'm like, they survived. They got to the doctor quick enough, and like once they exhibited symptoms, and they survived, but it can be extremely deadly. And I'm like, that there was a huge mandate for everyone to get tested and get vaccines for it, that lived in the dorms. There were lines blocks long to get vaccines for it. Now, when I say, I'm like, it can be deadly, because once that bacteria gets into the cerebrospinal fluid, it can cause death within hours after you start exhibiting symptoms. It is fast. Luckily, those at UW Madison survived, but it's deadly and it works quickly. If it gets into the blood, it also can be life-threatening and deadly, but it usually takes at least a few days to cause death. Either way, neither of it is good. Now, it also can cause what's known as petechia, is that this bacteria, especially when it gets in the blood, it can cause the bursting of blood vessels, and so you get these hemorrhagic lesions that are visible on your skin. Now, this, of course, is worst case scenario when I look for pictures, and I'm like, but you have bursting blood vessels on your skin, and as the blood vessels are bursting, your skin is no longer getting the oxygen that was in the blood, and so you actually start to have tissue death. And so, of course, this, is extreme versions, but yes, and I'm like, you have death of tissue because of the bursting of blood vessels. Now, diagnosis, and I'm like, has to be rapid. It's critical. And I'm like, again, it can cause death within hours after symptoms. And I'm like, or it can cause death within a few days. What they're looking for are gram-negative diplococci in the cerebrospinal fluid. You can even see it also likes to live intracellular. Anytime you see gram-negative cocci, if you're exhibiting some type of symptoms, it's an indication it's Neisseria, and depending on where it is, whether it's in the blood, whether it's in the cerebrospinal fluid, that's when it can be problematic. Now, treatment, intravenous antibiotics. Again, you need to get that high concentration into the blood right away, and the top treatment is penicillin. Your best bet is just to not get it at all. And so I'm like, the problem, asymptomatic carriers can't treat because we don't always know, so it's getting tested. Otherwise, getting a vaccine, especially if you're at-risk individual. You can get vaccinated if you live in the dorms. You can get vaccinated if you think you might be, next to someone that's had it. And I'm like, again, they vaccinated just about everyone at UW-Madison once they knew this was around. I'm like, there is a vaccine. They used to require this vaccine for incoming students that lived in dorms. They don't require it anymore. They just strongly promote it, which then usually students are like, I don't want another shot. And so they don't get it. There is a vaccine for it, especially for those that I think they're going to be at higher risk. Otherwise, and I'm like, they can do prophylactic treatment. And I'm like, they'll give antibiotics to individuals that have been in close contact with someone that's had it. And I'm like, if your roommate just went to the hospital because of this, they're going to give you antibiotics. And I'm like, they want to make sure that if you've been exposed to it, they're going to kill this bacteria before it ever gets into the blood or before it ever gets into the cerebrospinal fluid. Now, that's my gram-negative cocci. Now we move on to our gram-negative rods, which again, divided by oxygen requirement. And we're going to start tackling these left to right, starting with our facultatively anaerobic. Now, again, to divide our facultatively anaerobic, we're going to do an oxidase test, and we're going to start with our oxidase negative. Again, still a big group of bacteria that are here, but we'll at least start with these. And so these two families in our facultatively anaerobic contain the most human pathogens. If I go back to my table, this is all genus, this doesn't even include species. This is a lot of bacteria that can cause diseases in humans. And yeah, they're the two groups, the Enterobacteriaceae and the Pasteurellacea, and we do this oxidase test to divide the two, whether they're oxidase negative or oxidase positive. And there are a lot of healthcare-associated or nosocomial pathogens in this group. If we look, these are our gram-negative bacteria. These are all, and I'm like, this whole table here are causes of healthcare-associated or nosocomial infections in the U.S., and there is a whole slew of gram-negative, and guess what? Most of them are in the Enterobacteriaceae. Now, if you remember, entero has to do with intestines. A lot of these are fecal-oral. Again, everyone eats poop. A lot of them are fecal-oral, and I'm like, it's bad hygiene is how a lot of these get spread. And so I'm like, the Enterobacteriaceae, these are a lot of times normal flora found in a lot of animals and humans, and I'm like, they can get somewhere where they're not supposed to. They're also ubiquitous or found in water, in soil, in vegetation. And I'm like, a lot of times still because it was fecal contamination of these areas. And I'm like, it's spread fecal oral route. These enteric bacteria are the most common gram-negative pathogens that we find in humans. Now, this becomes an issue. I have 2 bacteria here. If we saw both of these underneath the microscope in our lab, we'd be like, they're gram-negative rods. The question is, are they the same bacteria in A and in B? Although they both look alike, they're not the same bacteria. Almost that whole table, I don't want to scroll all the way back, but almost that whole table are gram-negative rods. They're all going to look alike underneath the microscope. So we have to be able to, divide them down. And there are lots more tests that you can do for narrowing down these gram-negative rods. And we'll do a bunch of them in our lab as well. And I'm like, here's a dichotomous key for some tests that you can do for some of these enteric bacteria. And I'm like, we can see if they ferment lactose. We can see if they're motile. We can see if they produce hydrogen sulfide. We can see whether they can break down urea. We can see whether they can use citrate. Now, some of these tests we're going to do in lab. We're not going to do the gelatin, and we're not going to do the Voges-Proxker, but we are going to look at some of these other different types of tests. It's how we can start to break down some of our gram-negative bacilli because there are so many of them. Now, for the Enterobacteriaceae, and I'm like, to diagnose someone with one of these bacteria, they're going to culture and grow the bacteria out in lab. One of the plates they're going to use, and I'm like, is the MacConkey agar, because MacConkey grows gram-negative bacteria, so at least you know you're a gram-negative bacteria. But MacConkey agar is also a differential media, and I'm like, which means it can or cannot turn colors. Now, the big thing you're looking for in MacConkey, because people are like, look, it turns yellow. That's not what we're looking for in a MacConkey agar, though. On a MacConkey, we're looking to see whether it can ferment lactose, which will show pink colonies. And I'm like, so it's one of the big indications in grouping and narrowing down these gram-negative rods. One, is it a gram-negative because it grows on a MacConkey? And 2, can it ferment lactose like E. coli can, and you end up having pink colonies? Otherwise, we're going to do a whole slew of biochemical tests. They're usually going to be done in tubes. You're going to see tubes like this coming up in lab in a few weeks that we're going to be setting up, and they turn fun, pretty colors. And I'm like, but it helps us narrow these down. Now, we also have tests. We've got computers that can run some of these similar tests in a way faster manner that can do that in the lab. Treatment for most of these Enterobacteriaceae is just to let the diarrhea happen. When you have diarrhea, the idea is to get to that feces out of the body as fast as possible, and most of the bacteria leave with the feces. And so I'm like, diarrhea in itself is a treatment. And I'm like, otherwise, if they thinking it is bad enough, and I'm like, that is obviously life-threatening because diarrhea can be, they will give antibiotics as well. Your best bet is to not get any of these particular bacteria. And the best way, since a lot of them are spread fecal-oral, is good personal hygiene, washing hands. And I'm like, and proper sewage control, which luckily in the United States we do have. Now, our Enterobacteriaceae, because yes, they were still a big group. They're oxidase negative. We've narrowed that down, but they're still a big group. And I'm like, so we narrow them down, since there are so many of them, into 3 kind of subgroups. The coliforms, the non-coliforms, and what we call our true pathogens. Coliforms, any of the coliforms means they rapidly ferment lactose. So on a MacConkey plate, you'll see pink colonies, like E. coli. E. coli is a coliform. And I'm like, but there's others. A lot of these are normal microbiota, normal flora that become opportunistic pathogens. E. coli lives in us, normally doing some great things, but if it gets somewhere else, it becomes opportunistic. Now, a lot of times they test for coliforms in water. A lot of times in the summer, come summer, and I'm like, you're going to hear them talking that they did testing at the beach, and they said there's a high coliform count. And everyone's like, I don't know what that means. I'll do it anyways. Generally, when they're talking coliforms, they're talking the only way they come, the coliforms got there is from feces. It's a fecal coliform count. A lot of times it's E. coli that can become problematic, but if they say don't swim in the water in the Mississippi because the coliform count is up, that means the poo bacteria is up. Don't swim in the water, it's dirty. And definitely don't drink the water. Everyone eats poop. So that's my little note on the fecal coliforms in water. And I'm like, non-coliforms, these are just ones that do not ferment lactose. So they're not going to have pink colony as in a McConkey plate. And then we have our group of true pathogens, which means even healthy individuals would get sick and I'm like, would come down or get some disease that these particular bacteria can cause. Now, in lecture, we'll continue looking at the specific bacteria in each of these groups, but I'm just going to end it right there, kind of previewing where we're going to start in lecture, and I'll kind of do a quick review of it, but I'm like, we're going to end right there for this particular recording.